Since this method analyzes cells from solution, it can be used to study cells in a near-native environment. This includes analysis of patient-derived cell samples.
This technique requires little to no sample preparation of cells. Therefore, we can analyze individual cells online under ambient conditions, which allows us to explore cellular heterogeneity.
This technique can be applied to study liquid biopsies from a variety of different disease states, since it can analyze different types of cells directly isolated from patient samples.
To convert single-bore glass tubing into a tapered probe with a sharp tip, first place a single-bore glass tube into the clamps of a vertical pipette holder, centering the glass with respect to the heating coil, and tighten to secure the tube in place. The heating coil is comprised of an 18-gauge nickel-chromium resistance wire coiled around a metal rod 2.5 times. Set the glass tubing with temperature program 19.5. Set the solenoid plunger at 4. Trigger the solenoid to pull the glass tubing. This step creates 2 probes fused at the tip. Use tweezers to cut approximately 1 millimeter away from the tip of each probe, creating an orifice of approximately 10 microns in diameter at the probe tip. To bend the glass probe for easy coupling to the ICMP single probe SCMS setup, first set a pulled glass probe into the microforge. Position the tip approximately 3 millimeters above the platinum heating wire. Then, turn the heat on the platinum wire to 30% of the maximum temperature. Bend the probe approximately 45 degrees from the original position. Place the inverted microscope microinjector in 2-cell manipulation.